Hi, everyone. Arsalan Arif here with Endpoints News, and thanks for joining us today for Weird and Wonderful Case Studies, Real-World Solutions for Unexpected Bioanalytical Challenges. We're sponsored by Agilex Biolabs, Australia's largest and most technologically advanced bioanalytical and toxicology laboratory, and I'm so excited to introduce today's panel. Joining us, we have Dr. Yanglang Feng, the Chief Operating Officer for Door Biologics. Dr. Feng oversees the early R&D, translational research, clinical operation, and business development activities at Door Biologics. We also have Sharon Hanegraaff, the Vice President of Drug Development at Kinoxis Therapeutics. Sharon is involved in management of the preclinical and ADME toxicology programs, drug substance manufacturing, and regulatory submissions, as well as being closely involved in early stage clinical programs. We also have Yogesh Patel, the Senior Method Development Scientist at Agilex Biolabs. He's responsible for the design, strategic planning, execution of innovative techniques, focused on the development of intricate ligand binding assays. Also, Kirsten Wilcox, Analytical Services Manager at Agilex Biolabs. Kirsten is skilled at working with EMA and FDA guidance to support the development of both clinical and non-clinical validations and sample analysis. Now, if you have any questions, be sure to hit the Q&A button at the bottom of your screen. We've reserved some time at the end of this webinar to answer your questions. And now let's get started on our first case study discussion with Sharon from Kinoxis and Kirsten from Agilex Biolabs. Sharon, can you briefly tell us about your molecule and the non-clinical program?
Thank you, Arsalan, and thank you to Agilex for including me on this panel today. So to provide a brief background to Kinoxis, Kinoxis is a private Australian-based clinical stage company originally spun out of the University of Sydney in 2019 to develop novel, first-in-class, small molecule therapeutics intended for the treatment of substance use disorders and social dysfunction in neurodevelopmental and psychiatric disorders. Our lead compound, designated KNX-100, is being developed for the mitigation of symptoms associated with opioid withdrawal in patients with opioid use disorder. Earlier this year, Kinoxis commenced a first-in-human phase 1 clinical trial under a US IND are in planning stages for a second possible indication of agitation and aggression in psychiatric disorders. Kinoxis is backed by a consortium of sophisticated investors and has secured funding from the NIH, National Institute on Drug Abuse, to develop KNX-100 for opioid use disorder. In addition to our lead compounds, we have a number of compounds targeting the oxytocin receptor, which include partial agonists and positive allosteric modulators of oxytocin. So to provide background on our story, in the beginning of 2019, Kinoxis initiated a multi-study IND-enabling tox program evaluating their lead compound, KNX-100. We engaged the services of a large global CRO to conduct toxicological and toxicokinetic evaluations in both a rodent and non-rodent species, and in total commissioned 5 studies to be conducted under GLP. In March 2019, Kinoxis transferred an analytical method to the CRO and provided available physicochemical data to aid method development. In June 2019, the first of the in vivo studies commenced, and plasma samples were collected and stored pending analysis.
All right, Sharon. Well, thanks for that. But can I ask you, so did you face any issues and can you be specific about them?
Sure. Fundamentally, the CRO was unable to successfully develop either a rat or dog plasma method that could successfully be validated. Initially, they employed a standard off-the-shelf, commercially available chemical analog as an internal standard, which did not track KNX-100 or the metabolite. And it was not until more than 6 months later did they suggest introducing a KNX-100 analog or a stable label compound, which is recommended in both EU and FDA guidance documents. They could not achieve the accuracy and precision required to meet bioanalytical standards, and commented on particular issues with dog plasma, which was communicated to us as the problematic nature of KNX-100 in specific lots of plasma. They had significant issues identifying a suitable analytical range, and at one stage proposed 2 methods, a high range and a low range per analyte per species, which would have resulted in 8 analytical methods overall. In general, they had a random ad hoc approach to troubleshooting. Communication was limited, and no method development summaries were provided.
Okay, so what was tricky about the project, actually? So what do you mean by limited communication?
We discovered along the way that KNX 100 was unstable in plasma and to a greater extent in whole blood, and therefore required additional stabilisation methods. The CRO identified the instability, but were unable to resolve the issue. With regards to communication, we had weekly team meetings with the broader group that included the project manager and in vivo study director. Bioanalytical updates were verbalised on the call, but there were no written communication or development plans proposing next steps. In early 2020, a method development summary was requested, but the output provided unfortunately just highlighted that there was in fact no plan nor proposed solutions.
Okay, and so how long did this problem last for?
The problem lasted for over 12 months. I actually joined Kinoxis in early 2020, so it had been an ongoing problem for 9 months at that stage. My first request was for the method development plan I mentioned earlier, so that I could understand the issues and solutions that have been tried and failed. We introduced a methylated analogue of KNX 100 as an alternate internal standard and synthesised a deuterated stable label, which improved sensitivity to some degree, but we still could not achieve consistent outcomes.
Okay, so can we talk about any of the cost and quality implications? Can you talk about some of those?
Absolutely. We spent approximately $85,000 US on method development with no deliverables. The pivotal in vivo studies were delayed as we were unable to select dose levels without PK data to guide dosing. Our overall program, including the IND submission, was delayed by about 12 to 15 months. We continually had to advise our investors of the delay, which ultimately reflected poorly on both Kinoxis and the CRO. And we had aged plasma samples in storage that had been collected in July 2019 and therefore required additional long-term stability data to support that storage.
Okay, so continue. I mean, you mentioned some results. So how did this affect you?
As I said, I joined Kinoxis early in 2020, so the impact was primarily on my colleagues who had limited bioanalytical experience and were therefore relying on the expertise of the subcontractor, in this case, the CRO lab. I can tell you they were certainly frustrated and at a complete loss as to how to resolve the situation.
Okay, so continue. So what stage did you try to give up dealing with the problem? Is this where Agilex came in?
Yeah, so for us, it was actually a case of serendipity. In March 2020, Kinoxis were approached by Agilex for a meet and greet. We met with them and explained the 12-month method development challenge and communication issues we've been experiencing. They were understandably quite horrified and immediately agreed to help us out of the hole.
Okay, and so talk more about that. How did Agilex help you get back on track now? So
we signed a CDA immediately with them, and within 2 days, the head of R&D had reviewed all of the data and provided us with an assessment. We continued to work with them in a consulting role for a while as they helped us navigate the issues and provide guidance, which we shared with the CRO. In parallel, they initiated some method development work on KNX 100 in human plasma. They immediately identified the stability issues and applied a systematic approach to identify a suitable stabilising agent. At all times, their communication was clear and comprehensive, and in the event that I had questions, they were always available for a call. We received a detailed results summary within the first week of initiation and regular weekly updates thereafter. Within 2 weeks, the Agilex R&D team had achieved more progress than in the previous 6 months with the CRO's laboratory. Within 2 months of commissioning Agilex to assist Kinoxis, it was clear to us that we needed to withdraw the entire bioanalytical component of the project from the CRO and transfer to Agilex, which we did in July 2020.
Okay, so very interesting story about meeting the Agilex team. Now, Kirsten, let's loop you in at this point and ask what issues you faced when you were approached by the Kinoxis team.
Thank you. So the primary issues we faced when the Kinoxis team joined us in July 2020 was the collection parameters for the analytes. These were not able to be confirmed prior to collection, which is a little bit cart before horse when it comes to normal bioanalytical method development. We had the understanding that the 7-day samples are being collected as per standard parameters and the 28-day samples have been collected with additional precautions. So we had to work our validation in order to be able to suit both of those parameters. Another issue we faced was that the samples were aging every day that they weren't assayed. And that meant that we had the potential of having to do long-term stability for years if we did not stop the clock. So we worked really hard to get the method in a place to be able to perform that analysis and put that stop on. And finally, as mentioned by Sharon, the customer IND submission and program had been significantly delayed by nearly a year. So our focus was to be as quick as efficient as possible to bring this program timelines back on track.
All right. Well, Kirsten, introduce the team here at Kinoxis that helps you achieve those timelines.
Yeah, so here at Agilex, we do our best to introduce a small team for each program that we bring online. So we have the client team, which was Sharon, who worked with us, kept the communication open, and we kept in touch with her on a weekly basis. We had our R&D team. So as previously mentioned, our head of R&D, Jason Goy, helped with the initial review and got this project on track. And then that was handed over to Spencer Clark, who is as a principal method developer here at Agilex. And he worked with Sharon and myself in order to get the method development on track. And then finally, there's a project manager, which was myself, and I worked to get the methods validated to maintain the GLP integrity of the program to support the IND submission. So some of the challenges that we faced in regards to this program was the stabilization. As alluded to earlier, the stabilization of the KNX100 analyte and its metabolite in blood and plasma was a key challenge. We had to investigate many stabilizing agents in order to ensure which one would be most appropriate for plasma. We had to consider both the lab safety and the environmental impact of these stabilizers. So that was the key challenge to start with. We then had to investigate whether the stabilizer's presence was only required in plasma or whether it would be required in blood at collection. As mentioned before, the samples had already been collected without that stabilizer. So we had to consider whether that would impact and what actions we could take in order to be able to provide that stabilization. And then one of the last challenges with the stabilization is to ensure that our standards and QCs are representative of those stabilized study samples in order to give confidence in the reporting of the unknown results for those samples. The SIL versus analog is also the next challenge. We had to put into case time and cost considerations, which would provide the greatest confidence in the reporting of those unknown samples versus which would be the quickest and efficient to get the program back on track. And finally, once we'd settled on a stabilization method, it was how to ensure the chain of custody during stabilization for those samples, as the stabilization process required taking the originally corrected sample, stabilizing to create essentially a new sample. And we need to ensure that we provide confidence in that chain of custody to ensure that the result being reported from that new sample has genuinely originated from the original sample collected.
Okay, so how did you, once you addressed those challenges, then how did you get those timelines back on track?
So the validations were started almost in parallel. The dog was the first, as the development had shown that the species had potentially more challenges, as the matrix needed stabilization prior to extraction, while the rat needed stabilization during extraction. Once the first two dog accuracy and precision showed acceptable performance, we moved ahead with the rat, and we used any learnings from the dog validation to apply it to the rat validation. We also started this process to stabilize the dog samples because at this stage we had confidence in the method of stabilization. Once Agilex targeted a fit for purpose assay with all core validation analysis completed, and with my experience in GLP and support from my QA group, we ensured the GLP status of the program was maintained. We kept the sponsor and the external study director, who was responsible for the overall program, in the loop to allow them to make informed decisions about proceeding with analysis. We ran both sample analysis programs in parallel, dedicating 2 systems and up to 4 analysts due to the known type timelines. Where weekends were required, we supported this either to ensure the systems were running to perform or to perform extra extractions to keep those systems running. All of the contributing teams at Agilex were aware of the program timelines, and they had allocated book slots in all groups to meet the deliverables.
Okay, so what were the advantages for Kinoxis's and Agilex's teams after this collaboration?
Well, from Kinoxis's point of view, we were able to steer our program back on track. By early 2021, we had sufficient information to inform a pre-IND meeting with FDA, which was immediately followed by an IND submission later the same year. This in turn enabled us to initiate our clinical program, which is still in progress. We also experienced an overall cost advantage in working with the local CRO. primarily due to the application of an R&D tax incentive, which is applicable to research conducted in Australia. Being on the same time zone was also an advantage, as it allowed for a rapid turnaround of data and subsequent feedback. And overall, there was peace of mind, knowing that we could trust the data being provided.
So the shared time zone was a great benefit here as well. The live communication to be able to discuss actions as we progressed was invaluable to keeping the study moving as fast as it needed to be. The single point of contact was also very valuable. Sharon made herself fully available during all stages of the KNX 100 program at Agilex, and having that single point of contact for the program was greatly appreciated by the Agilex team.
Oh, great. So it's always so hard to collaborate on that between teams. So how did you actually do that together? Can you guys shed some light, Sharon and Kirsten?
Sure. I firmly believe that communication, along with the quality of the data, is fundamental to any collaboration. And in this instance, communication was clear and mutual throughout the program. Both the R&D team and later the GLP team were very easy to work with and always communicative. They continually accommodated and worked towards our corporate timelines, and overall, the project was managed seamlessly.
Yes, and understanding those required timelines for the program, which were clearly provided from Sharon throughout, were invaluable to Agilex to ensure that all the required resources along the path were booked in advance and ready to go. Regular calls and emails between the core team helped to manage live challenges, to be able to come up with solutions collaboratively to overcome and meet the overall program data deliverables.
All right, so what tips and lessons would you give our viewers today who are currently in the same situation that you were?
Don't always assume that all laboratories associated with global CROs are equal and effective. In our particular instance, we discovered retrospectively that the laboratory selected was relatively new to the CRO and was being used to overcome resource issues. They clearly did not have the required expertise to solve the challenging problems. Don't be afraid to scope smaller, mid-sized laboratories who may have extensive experience and often have the ability to offer flexibility to smaller biotech companies such as Canox's. And finally, don't be afraid to push back on the larger companies and switch to another vendor mid-program if you do not believe they are providing the service for which they were hired, as ultimately good quality data is critical for successful drug development.
And rescue studies, though challenging, provide Agilex the opportunity to be part of that solution. It allows us to hone our developmental skills, communication, and time management to achieve an efficient outcome. At Agilex, we understand the importance of all arms of a program to a sponsor's ultimate goal to introducing a drug to market to improve the lives of patients.
All right, wonderful. Well, thank you so much, both of you. But now let's hear from Dr. Younglung Feng and Yogesh about the pitfalls that Door Biologics experience and how a bioanalytical partner like Agilex Biolabs help troubleshoot their issues. Now let's start with you, Dr. Feng, first. Can you introduce to your clinical program, please?
Sure, and thanks, Excellent, for your kind introduction. And I also would like to thank Agilex for inviting me to share our story. And so, Door Biologics is a clinical stage biopharma in China. We have 3 different therapeutic candidates that are currently in phase 1 trial. And Door Bio has developed 4 technology platforms which help to drive our discovery pipeline. So right now, Huadong Medicine, which is a publicly traded biopharmaceutical company in China, holds 75% share at Door Bio, where an acquisition deal back in 2021. Our lead candidate, DR10624, is a first-in-class GLP-1 receptor, glucagon receptor, and FGF21 receptor triagonist. It has a long half-life to support weekly dosing. So at this moment, DR10624 is in phase 1 SAD study in healthy volunteers, and we will initiate a 12 weeks MAD study in metabolic syndrome patients in New Zealand.
Okay, so tell us what was tricky about this program, and can you expand on the complexity of this molecule and these challenges for the method development of the ligand binding assay in particular?
Oh, absolutely. So as I just introduced, DR10624 is a triagonist which contains 3 different domains and interact with 3 different targets. So naturally, when you think about it, it's a very complicated molecule. So in order for us to fully understand how this molecule behaves in human subjects, we need to develop a highly sensitive bioassay which can distinguish the complete molecule from partially degraded proteins. Obviously, dialysis 4 is the first-in-class biotherapeutic protein, and there's just no generic detecting reagent that we can use. Therefore, we have to establish a bioassay with customized reagents. So to achieve this goal, we discovered and developed 2 rapid monoclonal antibodies that specifically recognize the N-terminal peptide and the C-terminus recombinant FGF21 mutant, respectively. If there's any tiny compromise of either the N-terminal peptide or the C-terminus protein, the rapid maps will not be able to capture the molecule. So these 2 rapid maps helped us to further develop a sandwich assay that only quantify the complete and intact Dialysis-4. This sandwich assay is really robust and works quite well in assay buffer. However, the development and validation of this assay in human serum metrics can be quite challenging. We have to balance the sensitivity, selectivity, and stability. When we work with another bioassay CRO, we could not even repeat the sandwich assay in assay buffer. As a result, we could not proceed to the PK assay development and validation.
Okay, so how long did this problem last, and what did you try to do about it?
So when we worked with that bioanalytical lab, the problem lasted for 5 months. We tried to solve the problem by testing different reagents, lab supplies, and even plates. Unfortunately, none of them worked.
Okay, so what kind of cost and quality and timeline implications did it have for DOR?
Unfortunately, it cost us significant time, money, and efforts. We also have to postpone the sample analysis and assay meetings.
Okay, so are you willing to talk about how it affected you personally, all of this?
Yeah, I mean, frankly, it is quite frustrating and exhausting even when I recall it now. I have to invest a huge amount of my personal and my colleagues' time in endless discussion, research, and troubleshooting.
Okay, so what stage did you try to give up dealing with the problem and go to a different lab?
Right, so I think it's roughly around 4 months. We felt this might be a dead end, and we began searching for alternative biolabs in Australia. Agilex was actually recommended by one of our partners based on her previous experience. Now we feel it was the right decision. In the end, it never hurts to give it a try.
Okay, so how did Agilex's scientific team help getting your slipping study back on track?
Right, so when we contacted Agilex, we were impressed by the scientific team's experience and expertise. I also like Agilex business development team's professionalism and flexibility. We came up with a proposal that helped us to evaluate the assay in multiple steps. This definitely de-risks the collaboration and increase our confidence on Agilex every step of the way. I would also like to pass on to my partner Yogesh to expand on the solutions.
Yes, sure. So when I've been informed by my analytical manager about this assignment and got to know that the first obligation was to report the feasibility run within 1 week from the commencement of the project. Therefore, we have to adopt a cautious stage gate process with initial proof of concept to first demonstrate that the assay is behaving as expected and give sponsor the confidence in our technical capabilities and communication strategies with regard to the study. To be more concrete, we understood at first the context and the problem statement of DR10624 PKS and assess the science bottleneck previously faced with the method. As the DOR was more concerned about the sensitivity of the method, it was really challenging to precisely evaluate the attainable sensitivity where we had to start the development from the scratch. So we have defined a plan of action with detail about the reagent preparation and early optimization of the run, which focus on shaping the ELISA methodology for the molecule. And then we have started working on it along with setting up the weekly update, along with setting up the weekly call with DOR
Okay, so can you share what kind of assay platform is preferred? Any kind of challenges you had during the development phase, Yogesh?
Yes, the complex structure and mechanism of DR1064 molecule have led to different biological challenges due to the high potency and therefore low clinical doses of trispecific molecules. A very sensitive PK assay with LLOQ level, even at picogram level, may require. And for this kind of multi-domain molecule, method with high level of sensitivity can only be achieved with MSD platform. And that is why ECL-based MSD is more preferred and best platform. However, because of the longer lead time to procure MSD reagent at our site, we have started the development on more standard ELISA-based assay. And with the ELISA assay, we have successful to achieve LLOQ at single-digit nanogram level per ml, which also showed a very robust performance in initial development. But gradually, when we have encountered the selectivity issue at lower analyte level and also the short-term stability problem, which demonstrated under recovery of analyte subjected to the time exposure at room temperature.
Okay, so what was plan B?
Absolutely. As previously mentioned, that the lead time was completely longer for the MSD reagent, but we knew that this platform was the best for intended purpose. And that is why we planned for working on MSD in parallel to the ELISA platform. In fact, Agilex was able to translate to MSD method once reagent were received and also successful to mitigate selectivity issue by employing higher MRD with assay buffer on. To address the stability issue at lower level of analyte, we have introduced frozen calibration curve strategy and working on eyes, which has efficiently improved the outcome and thus allowed us to progress to the validation stage.
Okay, so what did Agilex do differently, both of you?
And I just go first. Agilex definitely understood our concerns on both timeline and cost. Although we are eager to solve the problem and get the sample analyzed as soon as possible, we still care about the overall cost. In addition, we have never worked with Agilex before, so this is our first project with Agilex. Naturally, we would still be cautious with the proposal, even we had great references on Agilex from our partners. When we communicated our concerns with Agilex, Agilex proposed this multi-stages plan that gave us the opportunity to evaluate the assay. We only need to pay for the next stage when the first stage is successful and complete. And do you want to comment more on that, Yogesh?
Yes. So having understood the urgency of the method, we have stayed with very close contact with the sponsor, followed by our regular updates through emails and weekly calls. In addition, development experiments were optimized in such a way to maximize the throughput in shortest amount of the time without compromising the quality of the data. I also want to draw your attention to the fact that the molecule is unique, that is first in class, and to have multiple domains present in that. And the choice of reagent and optimization were not straightforward, but it was effectively carried out with close collaboration with the Yongliang and Doyer team. Let me also highlight that the multiple resources were employed during method development. And to speed up the development activity, we have continued work even during the weekend to meet Doyer timeline.
No, weekends. Okay, so how is sample analysis planned based on the current scenario?
It's tough. Amazing, right? So Agilex has already analyzed our study samples, and that tells you how efficient this team is. We were also able to conduct the scheduled SRC meetings and presented the data, which is well received by the committee.
So let's talk about the communication process. How was it during this collaboration? Close, I'm assuming.
Oh, it's amazing. The communication with Agilex is just simply amazing. We have frequent and on-time email exchanges. There is a regular weekly meeting, and Agilex team is also willing to jump on a meeting with us if there is an emergency or as we need. We will also receive a very nice summary and the minutes after every meeting, which have been super helpful for our project management and internal control.
That's wonderful. So has the Agilex team helped rescue this study for Doyer? So what were the advantages for both companies? Help us walk through that, both of you, please. Dr. Fung first.
Sure. Since our lead candidate, DR-164, is first-in-class triagonist, which has huge potential for patients with severe metabolic diseases. From my own perspective, rescuing this study helped us to speed up our clinical trials, which is critical for drug development in a highly competitive field. I guess in the long run, this can also help to stand out from the competition in the field. And I hope ultimately the success of the clinical translation of DR-164 into the market. And you guys, do you want to add on that?
Yes, it's never too easy to rescue a study because timelines are tighter and challenges are bigger. Additionally, as previously mentioned, the singularity of trispecific molecule was also a unique complexity that we have handled. It was really satisfying to help sponsor to meet the timeline of this critical clinical study. I would like to expand on what Yongliang said previously, that we have stayed close contact with the sponsor with regular updates through email and weekly calls. We ensured that representatives from the method development, regulatory, business development, and operational team will be present to address all relevant questions. All our email updates include detailed spreadsheet with data table, experimental design, conclusion, next step forward, and recommendation. We were always flexible to jump on the call if required, even though there was a time difference and the participants were spread over 4 different geolocations. This was not a barrier because organization made it sure that there was still communication live.
Biopharm is a global industry. That sounds like a wonderful story, and I love to hear such a good collaboration story. So tell us, the viewers, some lessons learned from this experience.
Sure. From my own experience, finding a reliable bioanalytical lab can be one of the keys to a successful clinical trial. Also, I think communication and mutual trust are important.
Well, smartly selecting the reagent and detection so the intact form of multi-domain therapeutics can be detected, choosing the compatible platform, and even work on 2 efficient platforms simultaneously in order to have backup at a very advanced stage if required to change.
All right. And so now this is sponsored by Agilex Biolabs. So I'm going to ask you, any tips for our viewers and sponsors who are currently in the same situation as you were, Dr. Fung?
Sure. I mean, I would recommend working with Agilex for sure, or at least give them a try.
As Agilex Biolabs unfolding new avenues across the globe and expanding its strengths and resources to anticipate world-class bioanalytical services, we are expecting more challenging studies and projects. And what could be more exciting than to work on rescue studies? In Agilex, we are aware of the significance of the stage of drug development in achieving sponsor ultimate objective to bring a medicine to the market for better the life of patients. So please start discussion with us as early as possible in your drug development program.
Well, thank you. Really, that was a wonderful discussion, and on rescue studies. Really, it's a very important thing, and I really thank Agilex Biolabs for sponsoring this and bringing this. So let's get to a few questions additionally before the end of our webinar. So Sharon and Kirsten, I want to ask you, how are such tight timelines on sample analysis? How was that manageable?
I can speak to that first. So again, comes back to communication, constant communication, and clear guidelines around timelines and requirements. Agilex worked beautifully with our CRO because the samples were being collected in the US at the CRO and shipped to Agilex. And again, that was all done very smoothly and very well handled.
And having the clear goals of what we needed to achieve and when we needed to achieve it. We worked well with the CRO in the US to get waybills in advance. We could then track them through the courier system, get our resources all booked. As I said in the previous conversation, we would had all of our groups booked. So our sample management team, our quality group, our analytical team, they all knew about the importance of this study. All of them were ready to go when the samples arrived. So that really helped to get everything on track. And Sharon kept us on board with all of the changes and all of the things that were upcoming. So we were always able to sort of course correct in a live manner.
Wonderful. So let's talk about some of the stability issues that you encountered. So explain actually in detail how Agilex overcame these issues when the previous CRO could not.
So we observed low recovery from plasma across all species, but in particular the dog, which demonstrated rapid degradation, which then put into question the stability of KNX 100 at the point of collection as well. Agilex immediately investigated a wide range of potential stabilisation agents under both controlled and stressed conditions over time. In parallel, Agilex and Cynopsis were working collaboratively on exploring published literature, which identified an antioxidant that had been previously used for stabilisation of a similar compound. Over time, as more information on our compound came to light, improvements were made to the stabilising agent, and the method was optimised accordingly.
Okay.
Yeah, so
Kirsten, please.
Yes, so we worked, like I said, closely with Sharon and Spencer, Spencer Clark, who was our lead developer on this, was reading all of the available literature, checking up on all of the stabilization techniques. He kept me fully informed of his progress, which allowed me to then take that forward into the validation and adapt the experiments in order to ensure that they met the criteria without any surprises. So it was working all together as a group that really helped us to get that stability issue under control, and has now led to an optimized method, which we're using in our human trial.
Great, thank you. Now, Dr. Fung and Yogesh, I'd like to ask about the multi-stage approach and why that might be helpful for a sponsor. Can you give us some insight into that?
Oh, absolutely. And by the way, that's a very interesting question. So when we talked with Agilex, we have agreed on this multi-stage approach. So the way we agreed on is, since PK assay development and validation is actually a multi-stage steps, we need to think about the first and make sure Agilex can repeat the assay in assay buffer, and then if Agilex is successful in developing the assay, we will then move to the assay validation process. So in that way, we don't need to pay for everything upfront, and we are not actually bounded by the contract for the full suite assay development and the validation. We're only paying for the next step when the previous stage is successful. So that helps us to both lower down the risks and increase the confidence every step of the way.
Thank you.
Definitely, it's like I just want to add up on that strategy where we are using our resources on multi-stage approach, and we are not using all our resources at a time, but we evaluate all the assay reagent and everything in such a way that it will give us the confidence and also the sponsor that we can go ahead with the study and then we will get the success.
Right, wonderful. Well, that's all the time that we have for today. I want to thank everyone in the audience for tuning in, and I want to give a special thank you to all of our panelists for sharing their time today. And I appreciate Agilex Biolabs for sponsoring this discussion. If you would like to rewatch this webinar or share it with your colleagues, a link for on-demand viewing will be sent tomorrow. I'm Arslan Arif for Endpoints News. Thank you for joining us, and we hope to see you at a future Endpoints webinar.